I'm the research manager at the Macular Society, and I'm hosting this webinar this evening. Tonight's webinar is suitably on one of our research projects. We have about 30 research projects on the go at any one time. And it's going to be on isoflavonoids, which are a group of chemicals from the hyacinth family, and the possibility of them being a new treatment for wet AMD. Now, we've got Professor Tim Corson here, speaking all the way from Indianapolis, and I'll introduce and tell you a little bit about him in a moment. But to remind everybody that Tim will give you a talk for about 25 minutes, and then we'll open questions. Please put your questions in the chat for any time during the course of the webinar, and then we'll have about half an hour after Tim's given his talk to answer lots of questions. And I'm sure there will be plenty of questions on new treatment for wet AMD. So I'm going to therefore introduce our speaker tonight, Professor Tim Corson, who works at the Department of Ophthalmology at Indiana University School of Medicine in Indianapolis in America. He holds a BSc, an MSc and PhD degrees from the University of Toronto, and he completed a postdoctoral fellowship in chemical biology at Yale University before establishing his laboratory group at Indianapolis. His laboratory focuses on chemical biology approaches to target identification and drug discovery for wet AMD and ocular tumours. So he works with Professor Dulcie Mulholland at University of Surrey on this research project, and that may strike a chord with some of you because we featured Professor Mulholland and this project as part of the Macular Week appeal in June. So you may recognise this as being the Hyacinth project, as we called it. So I'm now very much happy to welcome Professor Tim Corson, who's going to tell you all about his research project, which is a 3-year research project, and it's about halfway through, maybe about a year into its 3 years. Tim, welcome.
Thank you so much, Geraldine, and good evening, everybody. It's my pleasure to be here tonight through the magic of Zoom. I'm sorry that doesn't mean I get to travel to the UK, but it means I can sit here at home on a Tuesday afternoon and talk to you. So yes, we are very thankful for the support from the Macular Society for this really exciting project that we're working on. But I should say, Geraldine, can you see my slides?
Yes, we can.
Okay, wonderful. So we are interested in discovering new potential therapeutic leads for macular degeneration, specifically the wet form. As you heard, I'm based at Indiana University School of Medicine, but this project is really driven by my collaborator, Professor Dulcie Mulholland, who is a professor in the Department of Chemistry at the University of Surrey. And we've worked together for a number of years on related projects. Before I jump in, I'd just like to point out how global research is, especially in the case of our collaboration. So I'm based in Indianapolis, which is just south of Chicago in the USA. And of course, Professor Mulholland is at the University of Surrey in Guildford, just outside London. But I'm originally Australian from Hobart, Tasmania, and Professor Mulholland is South African from Durban. And of course, we've traveled around to develop our research in our labs, and we've established this transatlantic collaboration, which I think is really indicative of some of the excitement in science these days, that it really brings people together from vastly disparate geographic areas and makes the research that much more fun. So I'm required to share a couple of disclosures with you. I'm a co-inventor on patents related to some of the work that we're presenting, and I do receive research support from, and I'm a consultant for a company called Evergreen Therapeutics. And perhaps most importantly, I am not a physician, so I cannot and should not give you medical advice, but I'm more than happy to answer questions about the biology of wet AMD. So in today's short talk, I'd like to give you a bit of a disease overview about some of the underlying biology of wet AMD and why and how we think we can target this. Then talk a little bit about our drug discovery projects for this disease, and then close by putting this in context with the drug development process and how we go from what we're doing in the lab to a drug that could be used in patients. So I like to think about eye disease and disease more generally in terms of death and growth of cells. Too much death is bad, and too much growth can be equally bad. So we think about too much death in diseases like glaucoma, where there is degeneration of some of the neurons in the retina, or other retinal degenerative diseases, including dry AMD, where the retinal pigment epithelium and the photoreceptors die when they should not. On the other side of the scale is diseases characterized by too much growth. Of course, the obvious example is cancers, where cells grow out of control in various places in the body, including in the eye. But this also happens in diseases like neovascular or wet AMD and proliferative diabetic retinopathy, where new blood vessels grow where they shouldn't normally. So blood vessels are a really important part of the eye, and I'm sure everyone is familiar with the basics of ocular structure. So light enters through the cornea, passes through the lens, and travels to the retina, which is the light-sensing tissue at the back of the eye. So the retina, shown blown up here on the right, has a blood supply within it, the red blood vessels that you might be able to see here. And these blood vessels supply the neurons of the retina. The light-sensing cells, called the photoreceptors, are some of the most energy-demanding cells in the entire body. So it's really important to have a good blood supply that provides oxygen and nutrients to those cells. And then the outer retina is also supported by another blood supply from a tissue called the choroid, which underlies the retina. So the retinal blood supply is absolutely essential for normal ocular function, but if blood vessels form where they're not supposed to, this can lead to a variety of eye diseases. So the formation of blood vessels is a process called angiogenesis. This is the process of formation of new blood vessels from pre-existing ones. So it's a sprouting process where a blood vessel sends out a sprout and develops a new vessel over time. And this is a very important physiological process. It occurs during normal health, especially in embryonic development. As you can imagine, as a fetus or an embryo is developing, it needs to develop its initial blood supply. It's also important in wound repair. If you cut yourself and a blood vessel is damaged, that vessel needs to regrow as the cut heals. And it's also an important part of the female menstrual cycle as the endometrium forms each month. But angiogenesis can also be associated with a variety of diseases. Tumor growth is one example. As cancers grow large, they need nutrients, so they will recruit blood vessels to help them grow and develop. But angiogenesis is also a feature of a variety of blinding eye diseases. So this process, which we call neovascularization for new blood vessel growth, is a key feature of several different eye diseases. In premature infants, it's a feature of retinopathy of prematurity. In adults, it's a feature of wet age-related macular degeneration, not the dry form. So the dry form of macular degeneration involves a slow and gradual degeneration of the photoreceptors and the retinal pigment epithelium. But when the disease transitions to the wet form, new blood vessels are growing and they are leaky, hence the name wet AMD. Neovascularization is also a feature of proliferative diabetic retinopathy. Again, this is one form of diabetic retinopathy, the most severe form, where new blood vessels form and can leak throughout the retina. And again, as with tumors elsewhere in the body, anytime a tumor grows, it needs to recruit a blood supply. And the image you can see here shows a retinoblastoma tumor, which is a childhood eye cancer, recruiting some blood vessels as it grows, looking very angry. So focusing in a bit on wet AMD, what actually happens? Well, the new blood vessels that form abnormally in this disease are abnormal. So they leak. They can leak fluid into the retina, which can upset the structure and shape of the retina. And in severe cases, they can even hemorrhage. So not just leaking fluid, but actually leaking blood in between the retina and the choroid. And this can lead to retinal detachment, which is very serious, but also leads to death of the photoreceptors and the RPE, leading to quite rapid and sometimes irreversible vision loss. AMD is increasing. I'm sure everyone is aware that our population across the Western world is aging, and age, as the name suggests, is the largest risk factor for AMD. So I'm showing some statistics here showing that in 2020, for the first time in the United Kingdom, the population of people over 65 years became greater than the population of people younger than 15 years, and this is expected to increase over time. If we look at the statistics in a different way, you can see the population 65 and older as a percentage of the overall population is increasing dramatically, and is expected to hit nearly 25% within 25 years. So as our population ages, we're going to see more diseases of aging like AMD. Luckily, as I'm sure many of you are aware, we do have some very powerful and effective drugs for wet AMD. And these are the anti-VEGF biologics. VEGF stands for vascular endothelial growth factor. And this is a signal that cells produce to basically say, hey, please, blood vessels, grow over here. So if we can block VEGF signaling, we can very effectively block new blood vessels from forming because the blood vessel cells do not receive that signal telling them to grow. So there are a series of drugs that are used for wet AMD, and I'm showing them here. Some of them you may be familiar with, like Lucentis, Eylea, Avastin. A couple of more recent additions are Beovu, and the most recent is Vabysmo, which targets not only VEGF, but another pro-angiogenic signaling pathway as well. As I'm sure many are aware, all of these drugs need to be injected directly into the eye, what we call an intravitreal injection. And this is something that has to be done by an ophthalmologist. There is one exception, the very recently approved Susvimo, or port delivery system, which introduces the drug ranibizumab through a device that's actually implanted in the eye and releases the drug over a longer period. So at least in theory, reducing the frequency of injections. These intravitreal injections have to happen everywhere from every month to every few months. So it is a significant burden on patients. Early in the pandemic, when we were all stuck at home, I had a couple of med students who had been working in my lab, and we had to come up with a project that they could do from their living rooms. So we decided to do a survey study of patients in our eye clinic, asking them their opinions of different delivery routes for retinal drugs. And in perhaps the least surprising research finding ever, we found that 80% of patients would prefer eye drops or tablets to intravitreal injections. So given this, we really think, although the anti-VEGF drugs are highly effective for most people, there is a critical need for new targets and new treatments that could perhaps be formulated as tablets or as eye drops to ease the burden on patients and the healthcare system as well. So that leads into the kind of work that we do in my lab. We're interested in finding potential new therapies. And we're focused on small molecules, what you would call chemicals or chemical compounds, rather than some of the big molecules like proteins that have been the focus of the existing approved drugs, because small molecules are more easy to deliver as eye drops or as tablets than those large molecules that really need to be delivered close to where the body is going to use them for them to be effective. So there are really only two ways to get new chemical compounds. Either we find them in nature, so-called natural products, or we make them in the lab through synthetic chemistry. So I'll talk a little bit about both these approaches because I think they provide some context for the kind of work that we do. So let's talk about natural products first. Natural products have for centuries been a source of some of our most effective drugs, such as aspirin from a willow tree. But these days, natural products tend to come from three main sources. There are microorganisms, so bacteria and fungi for the most part. The example here, the classic example that I'm showing is the penicillium mold that produces penicillin, which was the first and still widely used antibiotic available. So microorganisms produce some very interesting chemicals. Marine species are another very rich source of natural products. The image I'm showing here is a marine sponge that produces a cancer drug called Ara-C. And then plants are another very, very rich source of interesting compounds for all kinds of purposes. The example I'm showing here is the Madagascar periwinkle. It's a beautiful plant, and it is the only source of two very widely used cancer drugs called vinblastine and vincristine, and is a great example of a plant that produces something that really isn't found anywhere else in nature. So in Professor Mulholland's lab, they focus on isolating interesting chemical compounds from plants. Specifically, as you heard in the introduction, they're interested in a class of compounds called homoisoflavonoids, which is a very technical name, but it just describes a small group of compounds. And these are commonly found in members of the hyacinth family, the Hyacinthaceae, and also some related species. So one of the wonderful things about working with hyacinth species is that many of them are very beautiful ornamental plants. And here are some of the species that the Mulholland Lab has been working on recently. A couple of members of the genus Eucomus. These are also known as pineapple lilies because of the way they look with that tuft of leaves on top of the flowers. And then on the right, a plant called Scilla peruviana that is a member of the family commonly known as squills. And you may have seen some of these plants in your gardens or in botanic gardens. They are quite widely cultivated. So the Mulholland Lab has spent a long time working on these plants and isolating all kinds of interesting chemicals from them. They usually start with the bulbs of the plants because they are fairly easily obtained. They don't require growing of the plants, and they can be chopped up and ground up fairly easily to extract the compounds of interest. And so going from the bulb to the actual chemical is a fairly arduous process. So it involves separating all the different constituents of those bulbs. You can imagine there's some woody material that they need to get rid of, and then a gradual purification process for each of the chemicals. Now, the image on the left shows how it's done in the lab, and this may seem very technical, but basically it's separating based on physical and chemical properties. And if you have ever done something like make coffee or use vanilla extract, you're familiar with this kind of process. So in the case of making coffee, we use hot water to extract the caffeine and the flavor molecules from the other materials in the ground up beans and pass them through a filter. Or if you've ever made vanilla extract, you take vanilla pods, soak them in alcohol, and the alcohol extracts the vanillin and again other flavor compounds, leaving the bitter and unpleasant compounds in those pods. So basically in the lab, it's exactly the same process, just done with slightly fancier techniques and leading to purified compounds instead of interesting mixtures of flavorful compounds. The other way we find new therapies is by synthetic chemistry. So taking small existing chemicals and combining them in new ways to make molecules that have never before been seen in the world. And to me, this is tantamount to magic. I collaborate with Professor Mulholland and some other chemistry labs, and it's always incredible when we get handed a small vial with some white powder that we know is a chemical that's never before been tested for anything in the world, that didn't even exist a week ago. So I have a lot of respect for the chemists who have a variety of techniques that they can use to create new compounds from simple building blocks, as you can see in the pictures here. So once we have some interesting compounds, be they extracted from plants or developed in the lab, we do biological testing in my lab. So think about this as like a funneling process. We take a variety of chemicals and screen them for biological activity to try to find the ones that have some promising activity. The way we do this first is to look at the chemical's ability to stop the growth of blood vessel cells. So we use human retinal endothelial cells. So endothelial cells are blood vessel cells, and we have cells that are human in origin that will grow very nicely in the lab. And so our first screen is just to see whether any of these chemicals will stop the growth of these blood vessel cells. And we have a pretty neat way of doing this. We have a dye that we add to the cells. So the cells you can see growing here are spread out in these little wells of this plate. So each little square contains some cells, and we've added varying amounts of the compound and then added this dye that turns pink when the cells are growing and stays blue when the cells are not growing. So it gives us a really nice readout of whether our adding the compound has stopped those cells from growing. So this is a really beautiful assay. For those of you who are able to see the screen, can see it's pink and purple and blue colors. And it reminds me a lot of the unicorn frappuccino that Starbucks, at least here in the US, released a few years back, that contrasting blue and pink and purple color. Apparently the unicorn frappuccino tasted terrible, but I would warrant that our cells taste even worse. So what have we found? Well, we have some homoisoflavonoids, both derived from nature and also developed in the lab, that are pretty good at blocking the growth of those blood vessel cells. After we do that initial screen, we use some other assays. There's one here that I'm showing called the tube formation assay. If you grow these cells under just the right conditions, they'll form these tube-like structures that are almost like baby blood vessels. So on the left, there's an image of the control cells that form these beautiful tubes. And then when we add the test compound, that tube formation ability is really blocked. So this is really suggestive that some of these compounds are quite good at blocking the abnormal growth of blood vessel cells that we see in neovascularization in diseases like wet AMD. Some of the compounds we've found are quite potent and show some selectivity. So potent means that we can use a small amount to have a dramatic biological effect. And when we talk about selectivity, we're interested in finding compounds that will stop the growth of blood vessel cells, but won't indiscriminately kill other cells. You can imagine something that might be injected into the eye. You wouldn't want it to be toxic to other cell types. We really just want them to stop the growth of the blood vessel cells. So we do have some compounds that are really quite selective for the blood vessel cells. However, we do need to do much further testing before these are ready to move into human trials. And that leads me to the last component of my talk. I just wanted to remind you about the process of drug development. It is a slow and an arduous process, but this is important because it protects everyone from medications coming on the market that are not safe or not effective. So over the last 100 years or so, most countries have developed drug regulatory agencies. In the UK, it's the Medicines and Healthcare Products Regulatory Agency. In Europe, it's the European Medicines Agency, and in the US, it's the Food and Drug Administration. And these agencies evolved to make sure that if drugs are being given to people, they are A, likely to treat the thing that they say they will, and B, not likely to have side effects that are dangerous, or at least that there is a risk-benefit ratio of the effects, the efficacy to the side effects. So this drug development process is something that I think everybody's become a bit more familiar with over the last few years of the pandemic, as we've seen in real time the development of COVID therapies and COVID vaccines that have moved quickly, but still safely through this development process. So for our compounds, we're still at step one of the process, the discovery and development phase, coming up with the best molecules that have the best effects in the culture dish. And then they need to go through extensive preclinical research to make sure that they are safe, that they don't have unexpected side effects, and that they continue to do what we think they do. Then if they pass through all those stages, they can be approved for clinical trials, so taking them into humans for the first time. And as you may be aware, there are 4 phases of clinical trials. Phase 1 is an initial test in a small group of people to make sure that the drug is safe. Phase 2 is the first test to see whether the drug actually looks like it has the expected effect on the disease of interest. Then phase 3 is a big trial to make sure that effect on the disease of interest is still holds in a large population of people. And after phase 3, the drug will go for review by one of these regulatory agencies who will look at all the evidence, all the efficacy data, all the safety data, and weigh up, well, does the perceived benefit outweigh the risks of this drug? And if they agree that it does, they'll approve the drug for marketing. And then phase 4 trials occur after marketing when the drug is potentially being prescribed to tens of thousands of people. It's still important to look for rare or surprising side effects that may crop up over time. And drugs that have been on the market do get removed when surprising side effects are found along the way. So we are at this very early stage with our compounds. We are very excited, but there is still a long way to go. So to summarize what I presented today, I hope I've convinced you that plants are a very rich source of disease-modifying chemicals, particularly for wet AMD, but for other diseases as well. We have these homoisoflavonoids that can block blood vessel cell growth that is relevant to wet AMD, but extensive further work is required before we move to human trials. I hope that that you share my excitement, but at the same time, I don't want you running down to the end of your garden and starting to munch on hyacinth species. We're not at that stage just yet. I'd like to thank all the members of my lab that you can see here. I'd particularly like to point out with the 2 arrows, Alicia and Hannah. These are PhD students from Professor Mulholland's lab that came to visit us in Indianapolis. They're both chemistry students, and they were learning some of the biological techniques and sharing some of their chemistry knowledge with us, which was a really wonderful part of this collaboration. As I mentioned before, we're very thankful to the Macular Society for supporting this exciting project, and my lab is also supported by some other US-based charities. So thank you so much for your attention, and I'm excited to answer your questions.
Excellent. Thank you so much, Tim. That's a fascinating examination of the process you need to go through to find these new drugs. So please, everybody, we've got a few questions coming in, but into the chat function, and I'll give a minute or so. I'll ask one question and then we'll come to the chat function. I don't know whether other people are thinking this, but where did the idea from hyacinths come in, and what pointed you in that direction?
So that is a wonderful question. So Professor Mulholland's lab has been working on hyacinths for decades before she had any interest in working on wet AMD. When I first set up my lab, I found, just by reading the literature, a compound, one of these homoisoflavonoids, that had been shown to have blood vessel growth blocking effects. It was discovered by a group in Korea. So we started working on that compound and then expanded. And so I basically contacted Professor Mulholland out of the blue. I had seen that her lab had published a lot of work on these homoisoflavonoid compounds, and she was kind enough to respond to my email, and the collaboration has grown from there. So it started with a chance finding in the literature, and then this collaboration that's built since then.
Excellent. It's great to see collaborations happening across different countries. We're very proud of our research teams here, but I know there's a lot going on in America as well. Thank you. Right, so we've got a few questions coming in. So one question is, how will your drug be administered to the patients? I think you suggested it was not going to need to be an intravitreal injection.
So we hope that it could be delivered by eye drops or in tablet form. We're at too early a stage to know whether that's actually feasible, but that's certainly what we're aiming for, and one of the reasons why we focused on these small molecules that might be better able to pass through the structures of the eye as an eye drop if formulated in the right way. We do have some promising data with another homoisoflavonoid that we developed previously that did show some effects when administered as an eye drop in an animal model. So we think there is promise there, but it still really remains to be seen. But that's certainly the strategy we're aiming for, to give an alternative or a complement to the intravitreal injections.
Thank you. Yes, we've got some other projects that are looking at the possibility of eye drops that we fund ourselves. And I know talking to members at groups which I go to, they'd love to have an eye drop. It's a bit of a no-brainer, really, isn't it?
And of course, it's not necessarily mutually exclusive. So you could imagine an eye drop that is given alongside the anti-VEGF injections that might allow the injections to be spaced out more than they currently are. So combination therapies are definitely a possibility as well.
Oh, great. Okay. So somebody else has asked, stay on this theme, what time scale are you looking for possible drops? I mean, this is really early stages you're at the moment, isn't it?
Right, yes. So it's probably going to be a few years before we have anything that's ready to go on the market, especially since formulation as eye drops is its own chemical challenge. So getting something from an eye drop to the back of the eye is it's no small feat. So there's some formulation technology that's involved. As you can imagine, anyone who's taken eye drops, they get rinsed away by tears very quickly. So you have to have enough compound there that can get into the eye fairly quickly before that eye drop is diluted away. So it's not necessarily an easy challenge, but it is one that we're working on in collaboration with some other groups as well. And the goal would be to have a once or twice daily eye drop, which is something that hopefully most people would be willing to do. Although nobody likes eye drops, but if it's something that can preserve your sight, we're hopeful that people would be willing to do that long term.
Thank you. So somebody's asked, have you tried any other plants, or why have you got other projects going on that are looking at other plant-based chemicals?
Yes, so it's quite an active area of research. There are a number of compounds that have come from a variety of different plants, from fairly esoteric species through common species like citrus fruits that people have tested in various models. One of the challenges with natural products in general is that for a lot of them, we don't really know how they work. And this includes some of the homoisoфlavonoids, although we do have some fairly strong leads from our lab as to how they're working at the cellular level. Of course, it's much easier to develop something into a drug if you've got a pretty good understanding of what it's doing at the cellular level. If it's kind of a black box, it just raises some more questions about, well, do we think it's actually doing something specific, or is it a non-specific effect? So some of the compounds out there that people have tested, it's very hard to know exactly how they're working. But plants have been a very rich source of compounds that do show that anti-angiogenic blood vessel growth blocking effect. And some of them have progressed in even as far as clinical trials in the past. But not everything that makes it into a clinical trial, unfortunately, succeeds as a drug.
Thank you. Yeah, so there's a few more questions around the sort of the people sometimes think that a natural compound is a safe compound because it's natural, and somebody said, would there be any likely side effects of this new drug? So natural things can be toxic as well as non-toxic.
Yes, they absolutely can. So there are many natural things out there. Strychnine is a natural product. There are all kinds of deadly poisons that are found in nature. So it's very important to really de-risk, as we say, any of these compounds that we're working on. So we do a lot of toxicity studies in the lab, both within the eye and on the cell types we're interested in, but also looking at other systems and also biochemical assays to make sure that our compounds are not interacting with proteins or enzymes in the cell that are unexpected. So right now, we don't have reason to believe that there would be any side effects with our compounds, but again, we're still at a very early stage. So I certainly don't want to make any claims yet that they would be side effect free.
Okay, thank you. I mean, eye drops, somebody's mentioned that they sting. And I don't know whether that's the drug or whether it's the drop, the fluid that's in the drop. And so how about including a drop of anesthetic? I suppose that's a little further on in the process of developing a drug.
That's certainly an interesting idea. Yet nobody likes those stinging eye drops. It does depend a little bit on on what's in the eye drop, how it's formulated. It should be possible to make one for many purposes that doesn't sting. But a little anesthetic is maybe not too bad an idea, unless it causes side effects of its own, which is always the risk. But I do like that idea.
Thank you. So I'm going to go back up to the top. So somebody asked quite early on, once treated with injections, doesn't the wet AMD become dry? Now, I know you're not an ophthalmologist, but...
Yes. So that's actually an excellent question. So when it is wet or exudative, that means the new blood vessels are growing, they are leaking. That's where the wet term comes from. If the growth of those new blood vessels is stopped, then it is no longer leaking. It's no longer exudative, as we say, but it could always reoccur again. And as I'm sure many people know, dry AMD does not always turn into wet AMD. It's really only a fraction of dry AMD cases that develop into the wet form, but it is the wet form that can result in fairly rapid and sometimes irreversible vision loss. So it is very important to keep an eye, if you'll pardon the pun, on your symptoms. And if you see any changes, if you're a dry AMD patient, then it's worth talking to your optometrist or ophthalmologist in case it is transitioning into the wet form.
I think there's a common misunderstanding, mainly it's not the patient's fault. I think it's the way it's described in the eye clinics, that people are told, having treatment for their wet AMD, that they're told that their eye has dried up, and they interpret that as it converting to dry AMD, but dry AMD being another form of AMD which can coexist alongside the wet. But there's a wet doesn't turn into dry, if you see what I mean. But it's a common misunderstanding. Okay, so drops prevent scarring of the eye, which the injections cause. So this is the cumulative damage, scarring, sometimes it's called fibrosis, that impacts on the retina and can mean the injections no longer are effective.
Yes, so that is one of the real therapeutic challenges right now, is that fibrosis or scarring that occurs that is not blocked by the current drugs, and there are other drugs in development that are specifically aiming to improve that fibrosis. We don't know with our compounds. That's not something we've explored yet. So unfortunately, we just don't have an answer on that question, but it would be wonderful if we were able to develop something that could prevent that scar formation.
Yeah, I think there is some research going on it, but yeah, possibly not.
Yeah, it's a very active area of research right now.
That's good to hear. Somebody's asked about gene therapy. I don't know whether you know a great deal about gene therapy for wet AMD, but your opinion might be on that as an option.
Yes, there are some gene therapies that are being tested in clinical trials. Essentially, they work in the same way as the anti-VEGF drugs, except instead of injecting that anti-VEGF protein into the eye, the gene therapy causes your own cells to produce it. So it's like you're producing Eylea within your own eye. It's quite incredible that's possible. There are a couple of those therapies that are still in trials right now. I think it's an exciting area to keep an eye on. As you may know, gene therapy has really taken off in the ocular field. The first gene therapy to be approved for any indication anywhere in the body was approved for a form of retinal degeneration about 5 years ago. And since then, there's been a lot of interest in the field. So there's a variety of gene therapies being tested for a huge variety of eye diseases. I think it's a very interesting area to watch. And on that note, I'll take the opportunity just to really encourage people, if you're interested in getting involved in some of these newer therapies, to talk to your physicians about the potential for clinical trials. There's a lot of new things that are being tested for wet AMD, for dry AMD, for other degenerative eye diseases. And if you want to be on the forefront of potential new therapies, there are plenty of clinical trials that you may or may not be eligible for. And even if you're just interested in helping our understanding of disease, there's a lot of clinical studies that don't necessarily involve taking a brand new drug, but maybe looking at your response to one of the existing drugs or the natural history of the disease that helps us with understanding. So it is hugely helpful for the field, for future patients, if you're interested and willing to get involved in those kinds of studies.
Oh, yes, I completely endorse that. We do have a lot of people amongst our membership and our contacts who are very interested in research. And we are often supporting researchers to help recruitment for their clinical trials. not just clinical trials, actually. Our most frequent request is to sort of advertise surveys, and people are gathering information on how living with macular disease, as well as what patients' views are and things. So that's a really important type of research to be involved in as well, surveys as well as being involved in clinical trials. So I'll just take the opportunity to mention that we do have what we call our research participant database, which people can subscribe to, providing a small amount of information either on our website, or we have a paper form. And that really registers with us that you are interested in hearing about research opportunities. And if we have an opportunity we think you might be interested in, we can let you have that information. But we also advertise on our website, through our e-newsletter, through our Side View quarterly magazine, membership magazine. Thank you. Somebody's put the link in the chat. Thanks very much to our website form, to the research participant database. So a couple more queries.
I swear that was not a stage question on my part, but
I was trying to slip it in
these opportunities.
No, thank you very much. I would probably try and get that in anyway. So a couple more questions about gene therapy. How would gene therapy be administered?
So the gene therapies that are currently being trialed, they have to be injected subretinally. So it's an even more elaborate injection than the current intravitreal injections. So the intravitreal injections go into that jelly-like space in the center of the eye. The subretinal injections are actually injected below the retina, which is where they need to be for the gene therapy, which is carried by a special modified virus, can actually get into the cells that it needs to be. So it's a fairly significant procedure, but the big advantage is that it probably only needs to be done once, or at very least once every few years, rather than the regularity of the intravitreal injections. Again, not without its risks, but it's becoming more and more established for a variety of eye diseases. So there's a lot more expertise and confidence with the technique now.
Yes, it is early days. From what I've said, I think one of the trials, people have gone 3 years without needing any anti-VEGF injections, which is amazing. But the long term is really, I think, the question mark over gene therapy. How long would it last for? Would you need another injection? And what would be the long-term impact of the eyes themselves, the cells themselves making the anti-VEGF? It's really promising area for wet and dry AMD, actually. So same principle for dry. Okay, I'm going to ask one of my questions now, because we've run out of ones in the chat. So you talked about the process from laboratory research through clinical trials and so forth. So should the results of this project be successful, and you want to take the drugs forward, what would be your next stage as researchers to take this forward as a potential treatment?
So unfortunately, the part we're doing right now is the easy part. The next stage, getting all the preclinical data ready to be able to go to one of the regulatory agencies and show them our body of work to convince them that the potential drug is safe and effective, is a lot of work. So normally it requires either creating a company or partnering with a company who can raise the significant funds that are needed to do those extensive, especially toxicity studies, because there's a lot that needs to be done to really rule out any risks, anything unexpected that might happen with a compound before it goes into humans. And usually it involves testing in multiple models of the disease as well. So commercialization, it's usually the next step. And people may have heard of the so-called valley of death, that is this phase in between the initial lab work that develops a project and the actual marketing of a drug. There's a fairly long and very expensive phase where all this preclinical work needs to be done. Fortunately, both Professor Mulholland and I do have some experience interacting with industry partners. I do have another project that has successfully transitioned to that commercialization phase, and more work is going on to try and build that package for regulatory approval. So we're excited and optimistic that if we do continue to show success, we may be able to generate some commercial interest to do the heavy lifting to take the potential drug to the next stage.
Lovely, thank you very much. So we've got a question in the chat about supplements that can be recommended for people at risk of macular degeneration. So those will be the ARID supplements, age-related eye disease study supplements. I think they're more popular in America than they are in Europe, possibly. What do you think about supplements for AMD?
Yeah, so the ARID supplements, it's a sort of interesting blend of vitamins, zinc, and a couple of actual natural products, plant-derived natural products, lutein and zeaxanthin. And they work basically by being antioxidants. So one of the key predisposing features for dry AMD specifically is a lot of oxidative stress in the retina. And this is something that happens naturally with aging. There's more oxidative stress in the body. It's exacerbated by things like cigarette smoking. And it's an important part of our understanding of how dry AMD starts to form. So there was a very, very big clinical trial of this vitamin supplement called the Age-Related Eye Disease Study, or ARID study, in the US. And it did find that the risk of dry AMD progress early or intermediate dry AMD progressing to advanced or wet AMD was decreased somewhat by this vitamin supplement. So it is quite widely encouraged by ophthalmologists here in the US for patients who have intermediate dry AMD. But it's something I would encourage people to talk with their physician about. Obviously, before you start taking any kind of supplements, it's worth discussing with your doctor. But they can help you weigh the risks and benefits, whether this is something that might be helpful to prevent the worsening of the disease. It doesn't prevent the initiation of dry AMD, or at least we don't have evidence to show that. But at least in some patients, it may decrease the worsening of the disease. And in the absence of approved drugs for dry AMD, it is one option that is out there.
Thank you. Yes, we know a lot of people. It's something you can do for yourselves, but they're not available on prescription in the UK, so you do have to buy them yourselves. There's a number of brands available. Do ring our advice and information service if you'd like to know more. But they are relatively expensive. You have to buy them yourself. And but yes, I think they're pretty popular. A lot of people take them. I hope it's slowing the progress of their disease. So I have an interesting one here. Is it possible that plant therapies will allow new photoreceptors, rods and cones, to regrow?
That is a very interesting question. Yes. So the therapies that we're looking at, these homoisoflavonoids, we don't have evidence that they are what we call neuroprotective or neurostimulatory. We're really focusing on stopping those blood vessels from growing. However, there are many labs that are interested in trying to regenerate the photoreceptors that may have died, or the retinal pigment epithelium that supports them. And there are some very interesting chemicals out there that, at least in the culture dish and in animal models, can help preserve and even promote the growth of the retinal neurons. There's nothing, to my knowledge, at least, that has transitioned into humans yet in that domain. One area that has started human trials is the use of stem cells to replace some of the damaged tissue in the eyes, especially of dry AMD patients, replacing some of that retinal pigment epithelium. There are clinical trials to do that now, which is a very exciting area, basically reintroducing cells to replace those that have died. So it's, again, a hot area of research, but it's not something that we are directly focusing on.
No, people are very interested in the stem cell. I think it's probably the only treatment that possibly offers the hope of restoring vision one day. If they can replace the photoreceptors, which is a big ask, that would offer the potential to restoring vision. So I know a lot of people are very interested in the progress with stem cell research. And we had Professor Pete Coffey, who's a leading researcher in this area, who spoke at our conference, if anybody's more interested to know about the stem cell progress with stem cell treatments. I don't know whether you're going to be able to answer this, but somebody particularly says that they were diagnosed with macular degeneration. They took ARIDS immediately, but one eye went wet after 2 years and the other went wet 2 years after that. I was told to stop ARIDS. What can I take now? I'm guessing they were told to stop ARIDS because they were at such a late stage in the disease that it wasn't going to be potentially helpful for them anymore.
Yes, unfortunately, the ARIDS formulation decreases the risk of that transition from dry to wet. But once that has happened, I guess there isn't the evidence there to support continuing to take them. I don't have an answer, unfortunately, because there's so much variability patient to patient. I think it's definitely something worth speaking with your doctor about, whether there are any supplements that they would recommend. Probably anti-VEGF is part of the plan, but very much an individual situation, and really take advantage of the expertise of your ophthalmologist or optometrist who knows your eyes well, can see inside your eyes in a way that nobody else can, and can provide you the best advice.
Thank you. Yes, it's difficult to comment on particular situations. Okay, I'm going to ask another one of my questions now. I think you suggested that these drugs would be cheaper than the current anti-VEGF drugs. Can you explain why they would be cheaper?
We hope that they could be produced very cheaply, because although the synthetic chemistry that I spoke about seems like magic, when it's well optimized, it can produce fairly large amounts of chemicals fairly quickly. Whereas the existing drugs, they're biologics, so they're whole proteins in most cases that have to be produced in these huge bioreactors. It's a fairly large and involved process. Whereas synthetic chemistry is very well established technologies. It's possible to scale up to producing kilograms of compound, usually in a fairly straightforward way. And that's been reflected a little bit in the prices of the existing drugs, at least here in the US, where more people are paying out of pocket for their drugs. Some of the anti-VEGF drugs have been exceptionally expensive. And our hope is that a small molecule therapy could be produced more cheaply, and then that cost would be passed on to or that lower cost would be passed on to the payers, be it individuals or health systems.
Thank you. Somebody's asked about Stargardt disease and gene therapy for Stargardt disease. It's a little bit outside your area. I don't know what you can say. Anything about gene editing?
So there are a lot of really interesting gene therapy approaches being tested for a variety of retinal degenerations. Sort of the classic indication for gene therapy is a disease in which one gene is damaged. And if you can replace that damaged gene with a functioning version, at least in theory, you can reverse the disease. And this is actually one of the quite amazing things I find about some of the gene therapies that are being tested for these retinal degenerations. In many cases, it seems like all the machinery in the eye is just sitting there. The photoreceptors still have structure, all the connectivity is there. Just replacing that missing gene, producing its protein target, is enough for the system to start working again. It's really quite wonderful, and shows the ability of the retina to adapt in response to the introduction of these therapies. I don't know specifically about clinical trials for Stargardt's, but because it is a disease caused by single gene mutations, I'm sure there are some options that are being explored, at least in the lab, if not yet in clinical trials.
We have a project at Oxford in exploring this with Professor Robert McLaren, as well as some other projects that are looking more broadly at potential treatments that would treat a wide range of inherited retinal diseases. There wouldn't be gene specific, because if we have to produce a new treatment for every gene, that would obviously take a great deal longer than something that would address maybe a lot of inherited retinal diseases in one go. by going a different route.
Yes,
So
the gene therapy approach, of course, is very powerful, but there are several hundred different genes that are involved in retinal disease because it is such a complicated organ. So it's just impossible to go after every single one of them with a gene therapy approach. And most gene therapies, as you say, are targeting that one disease gene in that one disease. It's obviously would have a greater potential impact to have some kind of multi-disease or broad-acting therapy. And yes, there are lots of very interesting approaches to try and rescue some of the damage that or non-functioning systems that occur in some of these retinal degenerations.
All right, I'm just scanning through all the questions. I'm trying to check that I've addressed all of them. I don't think I've done this one. How often does dry macular degeneration progress to the wet form? I think it's more.
It's somewhere between about 5 and 15%, roughly. So the majority of cases do not, but when they do, it's something that you want to be aware of. So therapy can be started as quickly as possible. And in looking at AMD patients overall, only about 10% of all AMD patients have the wet form. So again, it's a minority, but those with the wet form are much more likely to have severe vision loss than those with the dry form.
Thank you very much. So, there's one more. Yes, somebody's asking about the Valida light treatment. I don't know whether you know anything about this. It's a form of light treatment for dry AMD that in the UK, I don't think it's licensed in America, but it is offered in a few places in the UK. And we've been having discussions with the company that produced this light treatment. Do you know anything about it, or I can say a little bit about it?
I only know what you already said, so
Okay.
maybe you can expand on that a bit.
Okay, so this is it delivers 3 wavelengths of light, and it's been assessed in clinical trials, and there's some promising results. But I think we don't really have time to go into that detail about that here. We have mentioned it, and it comes up, as a possible new treatment for dry AMD. But the clinical trial results are really just coming through now, so it's a little bit unproven at the moment. And we're running out of time, and there's a few more questions coming in. But yeah. Yeah, I'm sorry. I think we've run out of time. Lovely is that we could carry on asking questions all evening. So thank you so much, Tim. That was really amazing, and thank you for giving such a lovely, clear talk.
Well, it's been my absolute pleasure, and thank you for the opportunity. And best of luck to everyone. And keep talking with your physicians. Learn about clinical trial options that might be a good fit for you.
Thank you so much. Okay, yeah, we're coming up on the hour, so it just remains for me to say that next month's webinar on the 7 p.m. on the 15th of November is going to be focused on dry AMD, in particular the potential new treatments that are coming through for dry AMD, and they will both be administered as intravitreal injections. And we've got some researchers going to be talking about some work they've been done on the acceptability of dry AMD in intravitreal injection treatments. So 15th of November, 7 p.m, we'll be back with another one of our webinars. So it just remains for me to say thank you for all your engagement, all the lovely questions, and see you again next month. Thank you very much, everyone. Good night.